In this session, we are going to have a quick recap about the enzymes, their classification, mechanism of enzyme action, factors influencing enzyme action, mechanism and equation, derivation, Lineweaver-Burke equation, and double reciprocal proto-enzyme reaction, enzyme inhibition, competitive, non-competitive, and uncompetitive inhibition, along with suicide inhibition and feedback inhibition, vitamins, classification, structure, and functions, and all this related topics. So, enzymes are biological catalysts. Without catalysts, chemical reactions such as sucrose oxidation could not occur on a useful time scale, and this could not sustain life. Enzymes have extraordinary catalytic power, often far greater than that of synthetic or inorganic catalysts. They have a high degree of specificity. Biological catalysts, and their extraordinary catalytic power, often far greater than that of synthetic or inorganic catalysts. They have high degree of specificity. Their specificity is high for their substrates. They accelerate chemical reactions tremendously, and they function in aqueous solutions. In very mild conditions of temperature and pH. Enzymes are central to every biochemical process. Acting in inorganic sequences, they catalyze the hundreds of stepwise reactions that degrade nutrient molecules, conserve and transform chemical energy, and make biological macromolecules from simple precursors. Much of the history of biochemistry is the history of enzyme research. Biological catalysts was first recognized and described in the 17th in studies of the digestion of meat by secretion of the stomach. Chemistry of enzymes. With the exception of small group of catalytic RNA molecules, all enzymes are proteins. Their catalytic activity depends on the integrity of their native protein conformation. If an enzyme is denatured or dissociated into its subunits, catalytic activity is usually lost. If an enzyme is broken down into its component amino acids, its catalytic activity is always destroyed. Thus, the primary, secondary, tertiary, and quaternary structures of protein enzymes are essential for their catalytic activity. Enzymes, like other proteins, have molecular weights ranging from about 12,000 to more than 1 million. Some enzymes require no chemical groups for activity other than the amino acid residue. Others require an additional chemical component called cofactor. Either one or more inorganic ions such as Fe2 plus, Mg2 plus, Mn2 plus, or Zn2 plus, or a complex organic or metalloorganic molecule called a coenzyme. Coenzyme act as transient carriers of specific functional groups. Most are derived from vitamins. Organic nutrients required a small amount in diet. Some enzymes require both a coenzyme and one or more metal ions for activity. A coenzyme or metal ion that is very tightly or even covalently bound to the enzyme protein is called prosthetic group. A complete catalytically active enzyme together with its bound coenzyme and or metal ion is called holoenzyme. The protein part of such an enzyme is called apoenzyme or apoprotein. Called apoprotein. Finally, some enzyme proteins are modified covalently by phosphorylation, like glycosylation and other processes. Many of these alterations are involved with the regulation of enzyme activity. So let's learn about the classification. Many enzymes have been named by adding the suffix ase to the name of their substrates or a word or phrase describing their activity. This urease catalyzes hydrolysis of urea, and DNA polymerase catalyzes the polymerization of nucleotides to form DNA. Other enzymes were named by their discoveries for a broad function before the specific reaction catalyzed was known. For example, an enzyme known to act in the digestion of foods was named pepsin from the Greek pepsis, digestion. The lysozyme was named for its ability to lyse or break down bacterial cell walls. Still others were named for their source. Trypsin, naming in the part of the Greek train to wear down, was obtained by rubbing pancreatic tissue with glycerin. Sometimes the same enzyme has two or more names, or two different names have the same name. Because of such ambiguities and the ever increasing number of newly discovered enzymes, biochemists, by international agreement, have adopted a system for naming and classifying enzymes. The system divides enzymes into six classes, each with subclasses based on the type of reaction catalyzed. Each enzyme is assigned a four-part classification number and a systematic number, systematic name, systematic name, four-part classification number, which identifies the reaction it catalyzes. As an example, the former systematic name of the enzyme catalyzing the reaction, ATP plus D-glucose is equal to ADP plus D-glucose 6-phosphate is ATP glucose phosphaterase, which indicate that the catalyzes the transfer of a phosphoryl group from ATP to glucose. ATP plus D-glucose is equal to ADP plus D-glucose 6-phosphate. So we will be naming that as ATP glucose phosphotransferase, which indicate that it catalyzes the transfer of a phosphoryl group from ATP to glucose. Its enzyme commission number is 2711. 2711. The first number 2 denotes the class name, transferase. The second number 7, the subclass, phosphotransferase subclass, phosphotransferase. And the third number 1, a phosphotransferase with the hydroxyl group as an acceptor. And the fourth number D-glucose as a phosphoryl group acceptor. For many enzymes, first we will be putting a class name, next subclass, third number phosphotransferase with a hydroxyl group as acceptor in the fourth number, and glucose as the phosphoryl group acceptor. For many enzymes, a common name is more frequently used, in this case hexokinase. A complete list and description of the thousands of non-enzymes is maintained by the Nomenclature Committee of the International Union of Biochemistry and Molecular Biology, IUB system of classification, like oxidoreductase, transferase, hydrolases, lyases, isomerase, and ligases. Oxidoreductase, transfer of electrons. So let's learn about oxidoreductases are used about the transfer of electrons, hydride ions or H atoms. Transferases for group transfer reactions, hydrolases for hydrolysis reactions, and like lyases for addition of groups to double bonds, and isomerases transfer of groups within molecules to yield isomeric formations, and ligases formation of CC, RS, CS, CO, and CN bonds by reactions cleavage of ATP or other cofactors. Now let's learn about the mechanism of enzyme action in detail. The enzymatic catalysis of reactions is essential for living systems. Their biologically relevant conditions and catalyzed reactions tend to be slow. Most biological molecules are quite stable in the neutral pH, mild temperature and aqueous environment inside cells. Furthermore, many common chemical processes are unfavorable or unlikely in the cellular environment, such as the transient formation of unstable charged intermediates or the collision of two or more molecules in the precise orientation required for reaction. Reactions required to digest food, send nerve signals, or contact a muscle simply do not occur at a useful rate without catalysis. An enzyme circumvents these problems by providing a specific environment within which a given reaction can occur more rapidly. The distinguishing features of an enzyme catalyzed reaction is that it takes place within the confines of a pocket on an enzyme called the activation site. The molecule that is bound in the active site and acted upon by the enzyme is called the substrate. The substance of an active site is lined with amino acid residues with substituent groups that bind the substrate and catalyze its chemical transformation. Often the active site encloses a substrate, sequestering it completely from solution. The enzyme substrate complex, whose existence was first proposed by Charles Adolphe Woods in 1880, is central to the action of enzymes. It is also the starting point for mathematical treatment that define the kinetic behavior of enzyme catalyzed reaction and for theoretical description of the enzyme mechanisms. The surface of the active site is lined with amino acid residues with substituent groups that bind to the substrate and catalyze its chemical transformation. Often the active site encloses a substrate, sequestering it completely from solution. The enzyme substrate complex, whose existence was first proposed by Charles Adolph Woods in 1880, is central to the action of enzymes. It is also the starting point for mathematical treatment that define the kinetic behavior of enzyme catalyzed reactions and for theoretical descriptions of enzyme mechanisms. A simple enzymatic reaction might be written as E plus S gives ES and EP, or E plus P, where E, S, and P represent the enzyme substrate and product. EP and ES are transient complexes of the enzyme with the substrate and with the product. The function of a catalyst is to increase the rate of reaction. Catalysts do not affect reaction equilibria. Any reaction such as substrate to product can be described by the reaction coordinate diagram, a picture of the energy changes during the reaction. Energy in biological systems is described in terms of free energy G in the coordinate diagram. The free energy of a system is plotted against the progress of the reaction coordinate. The starting point of either the forward or the reverse reaction is called the ground state. The contribution of the free energy of the system by an average molecule under a given set of conditions. Energy in biological system is described in terms of free energy in the coordinate diagram. The free energy of the system is plotted against the progress of the reaction coordinate. The starting point for either the forward or the reverse reaction is called the ground state. The contribution to the free energy of the system by an average molecule under a given set of conditions. To describe the free energy change for reactions, chemists define a standard set of conditions: temperature 298 Kelvin, partial pressure of each gas 1 atm, or 101.3 kilopascal, concentration of each solute 1 molar, and express the free energy change for a reacting system under these conditions as delta G0, the standard free energy change. Because biochemical systems commonly involve H plus concentrations far below 1 m, biochemists define a biochemical standard free energy change delta G, the standard free energy change at pH 7. The equilibrium between S and P reflects the differences in the free energies of their ground states. In the example shown in the figure, the free energy of the ground state is lower than that of S, so delta G0 for the reaction is negative and the equilibrium favors product. The position and direction of equilibrium is not affected by any catalyst. So reaction coordinated diagram, there will be a ground state. Next, the ground state will be there, and free energy is plotted against the y-axis, and the reaction coordinate, that is the progress of reaction, is on the x-axis. Then the value reaches the maximum transition state, then lowers, and then just stops in the ground state. The free energy of the system is plotted against the progress of the reaction. This kind of diagram describes of the energy states during the reaction, and the horizontal axis reaction coordinates reflects the progressive chemical changes, bond breaking or formation, and S is converted to P. The activation energy is delta G. For the S to P and P to S reactions are indicated. Delta G is the overall standard free energy change in the direction S to P. A favorable equilibrium does not mean that the S to P conversion will occur at detectable rate. The rate of reaction is independent on the entirely different parameter. There is an energy barrier between S and P. The energy required for alignment of reacting groups, formation of transient unstable charges, bond rearrangement, and other transformations required for the reaction to proceed in either direction. To undergo reaction, the molecules must overcome this barrier and therefore must be raised to a higher energy level. At the top of the energy hill is a point at which decay to the substrate or product state is equally probable. It is downhill either way. This is called the transition state. The transition state is not a chemical species with any significant stability and should not be confused with the reaction intermediate. It is simply a fleeting molecular movement in which events such as bond breakage, bond formation, and the processes charge development are proceeded to make precise point at which decay to either substrate produce product is equally likely. The difference between the energy levels of the ground state and the transition state is the activation energy. The rate of a reaction reflects this activation energy. A higher activation energy corresponds to a slower reaction. The transition state is not a chemical species with any significant stability and should not be confused with a reaction intermediate such as ES or EP. It is simply a fleeing molecular movement in which events such as bond breakage, bond formation, and charge development have proceeded to the precise point at which decay to either substrate product is equally likely. The rate of reaction reflects its activation energy. The higher activation energy corresponds to slower reaction. Reaction rates can be increased by raising the temperature and or pressure, thereby increasing the number of molecules with sufficient energy to overcome the energy barrier. Alternatively, the activation energy can be lowered by adding catalyst. Catalysts enhance reaction rates by lowering activation energies. Enzymes are an exception to this rule that catalysts do not affect reaction equilibrium. Catalysts enhance reaction rates by lowering activation energies. Enzymes are an exception to the rule that catalysts do not affect reaction equilibrium. Any enzyme that catalyzes the reaction substance to product also catalyzes the reaction product to substance. The role of enzyme is to accelerate the interconversion of substrate and product. The enzyme is not used up in the process and the equilibrium point is unaffected. However, the reaction reaches equilibrium much faster than the appropriate enzyme is present because the rate of reaction is increased. So free energy will be there in the y-axis. Reaction coordinate will be in the x-axis. Then transition state can be seen. Then it lowers into ground state. In the reaction substrate to product, the enzyme substrate complex and enzyme product intermediates occupy minimum in the energy process curve of the enzyme catalyzed reaction. The terms delta G and catalyzed and delta G catalyzed correspond to the activation energy for the uncatalyzed reaction and overall activation energy for the catalyzed reaction respectively. The activation energy is lower when the enzyme catalyzes the reaction. Any reaction may have several steps involving the formation and decay of transient chemical species called reaction intermediate. A reaction intermediate is any species on the reaction pathway that has a finite chemical lifetime. When the S to P or reverse reaction is catalyzed by an enzyme, the activation energy is lower when the enzyme catalyzes the reaction. Any reaction may have several steps involving the formation and decay of transient chemical species called reaction intermediates. When the S to P reaction is catalyzed by an enzyme, the enzyme substrate and enzyme product complexes can be considered intermediates. Even though enzyme and substrate stable chemical species, the enzyme and substrate and enzyme and product complex occupy values in the reaction coordinates diagram. Additionally, less stable chemical intermediates often exist in the course of an enzyme catalyzed reaction. The interconversion of two sequential reaction intermediates thus constitute a reaction step. When several steps occur in a reaction, the overall rate is determined by the step or steps in the highest activation energy. It is called a rate limiting step. In simple case, the rate limiting step is the highest energy point in the diagram.